So hello friends, today we are going to discuss the antibacterial drugs. Okay, antibacterial drugs. Okay, so today we will discuss the vancomycin. Which drug we will discuss today? Vancomycin. Okay, so it is the drug which attack on the cell wall. Okay, so means it is the selective drug which inhibit the cell wall synthesis. Okay, so antibacterial drugs, the mechanism of action of vancomycin is uh, fish type. It attacks on the cell wall synthesis. Okay, so if cell wall synthesis will be break down, then what happen here? The bacterial growth will be reduced. Okay, means it kill the bacteria by uh, which mode of action it attacks on the cell wall synthesis, means it stop the cell wall synthesis. Okay, so if you see, suppose that we will see, suppose that it is the bacterial cell. Okay, so it is the, suppose it is the cell wall of bacterial cell. Okay, and the inner membrane is cell membrane. Okay, so it is the cell membrane. Okay, so it is cell wall, it is the cell membrane, and in this cytosol is present. Okay, and so if you see here, I will cut here one piece, one small piece, and we will see the chemical nature of this one. Chemical nature of cell wall part. Okay, so what happened here, if you see this one, so if you see the chemical nature of this part, it will seen like this. It will repeat again, so like that. Okay, so these fragments, these are the fragments. The name of this one, it is the, actually it is the L-alanine, D-alanine, means it is the polymer of amino acids. Okay, this structure is considered as a NAG. Okay, this is named as an a name. Okay, and this fibrous structure is known as glycosidic bond. Okay, again if you see in this part, these two units is called transglycosylase. What is the name? Transglycosylase. Okay, so suppose that if you see, what is this one, actually this is the full name is N-acetyl glucosamine, glucosamine, and this one is N-acetyl muramic acid. Okay, so if you see the chemical nature of this one, so simply if you see glucosa means it is related to glucose, means glucose will be present in this. N-acetyl acetyl group will present, amine part will be present, so three fragment is present in the structure. So if you see, it is the, okay, so it is the is, if you see this one, this is the fructose type sugar. Okay, and in this carbon, what happened, this is the amine part. Okay, and amine is joined together to through which part? Acetyl part. Okay, so this is the acetyl part of the NAG, and this is the amine part. Okay, this is the carbohydrate parts. Carbohydrate means it is called glucose. Okay, so glucose amine N-acetyl. Okay, N-acetyl, why it is called N-acetyl? Because it is attached to nitrogen. Okay, the remaining part is if you see one OH will be present here, this one. Okay, so this is the simple structure of N-acetyl glucosamine. Okay, so N-acetyl means this is the acetyl part, N-acetyl card because this is attached to N. Okay, acetyl is this one, glucosamine, this is the glucose structure, amine part is attached, so it is called N-glucosamine. Okay, so similarly if you see the which structure, muramic acid. Okay, N-acetyl muramic acid. So the difference between these two is only in this OCH3 present and in this OH is present in this position. Okay, so it is the chemical nature, means structure of NAG and name. Okay, so what happened here, if you see vancomycin, suppose that this one, this is vancomycin. Suppose that if any patient, when vancomycin is given, so what happened here, it will not affect these parts. Okay, means it name NAG and N-acetyl, D-alanine and this is the amino acid, this is NAG, it will not affect, means this part will be not affected by the vancomycin. There will be no effect on the glycosidic bond and NAG and name, and remaining is D-alanine, so it will affect only here. So it targeted the trans glucosamine, and what happened here, it will attached here, this one. Okay, so it will stop the attaching transglycosylase in this part. Okay, and itself it attached in this part. Okay, so it blocks the function of what you can blocks the function of transglycosylase. Okay, so because of it inhibit the transglycosylase, so there will be a dysfunctionation of transglycosylase, which is the important part of cell wall synthesis. Okay, so that's why cell wall synthesis will be stopped here. Okay, so by which, if cell wall synthesis will be stopped here, means it kill the bacteria, if bacteria will be killed, then the drugs which kill the bacteria is called bactericidal. Okay, so the drugs is called bactericidal drugs. Okay, so it is the simple mechanism by which vancomycin act. Okay, so this is the only I have discussed today, vancomycin. Okay, so if you see here, one part is we will also discuss here. Suppose that this is the bacterial cell, the inner is the cytosol. Okay, okay, so in this, the inner part containing the high osmotic pressure. Okay, so the inner part feels the high osmotic pressure, and it will bind to the water, means it capture the water. Okay, so this is the thick, cell wall is the thick. Okay, cell wall is the thick. Okay, so if cell wall synthesis will be stopped here, this will change high osmotic pressure, change to low osmotic pressure. Okay, so here what happened here, osmotic burst. Okay, so it is the simple mechanism, but you should remind this part. Okay, this part is very important, and if you because there are lots of bactericidal, or you can say the drugs, antibacterial drugs which act like that. Okay, so when I have discussed only vancomycin, but in this mechanism several drugs follow these rules, means it will attack on the glycosylase. Okay, so it is the one. So thank you.